All right, this is lecture 14 in microbiology. We're going to talk about the principles of disease and epidemiology. Now, disease is an abnormal state in which the body function is disrupted. It's not normal. Pathology is the study of disease. Etiology of a disease is the study of the cause of that disease. What is a disease is etiology. What is the etiology of this? Means the question is, what's the cause? Epidemiology is the study of the distribution and causes of a disease or some other health-related event. So where is it occurring? What is causing it? How many cases are there? Where are they? What's the underlying cause? What's the reason for it? Things like that. Pathogenesis is the mechanisms that are involved in the development of the disease, and infection is an invasion or colonization of the body by pathogens. So each of these has a different meaning, and I'll use these terms, and you need to understand them. Now, it's very important that you understand there's normal microorganisms living on the surfaces of the body, and those include external surfaces and internal surfaces. So you do have microorganisms that are present and are normally present and need to be present on your external epithelium, your skin, stratified squamous epithelium, but also on your internal surfaces, which are your mucosal epithelium, and that includes the GI tract, the GU tract, the respiratory tract, and they perform actually quite an important role. They're permanently there, colonizing an individual host, and they act to protect the host because they'll occupy spaces or niches that pathogens might otherwise occupy. Some of them produce acids which inhibit growth of pathogens, and others produce molecules that I wouldn't worry about the particular name here, but molecules that can inhibit the growth of other pathogens, other organisms that are pathogens. Sometimes you have, instead of some of those microorganisms that live on you may not be permanently there. It may not be a case of permanent colonization, but could be transient. So those are called transient microbiota. They're there for some time, but not 100% of the time. So this is just a diagram that shows, places where you could find normal microbiota on the inner surfaces. And by inner surfaces, I mean epithelial, mucosal epithelial surfaces. All these epithelial surfaces act as a boundary or barrier between you and the outside world. There are a number of different relationships that could exist between the normal microbiota or microflora and the host. There are a number of, they're all symbiotic relationships between the two, the organisms, the microorganisms, and the host. There can be different types of symbiotic, not symbiotic, but symbiotic relationships. There's commensalism, in which one organism benefits and the other is unaffected. There's mutualism, in which both organisms benefit. There's parasitism, in which one organism benefits at the expense of the other, and that's not the case for normal microbiota, because that would not be a normal situation. And then there's antagonism. These are different types of symbiotic relationships. We have with our normal microbiota, we have either commensal relationships or mutualistic relationships. So mutualism or commensalism, either both benefit or one benefits and the other's unaffected. We do not have these kind of relationships with normal microbiota. Now, one of the roles of the normal microorganisms that are living on your surfaces, external or internal surfaces, is that they act as competitors with other organisms that might be pathogenic. So there's always a competitive relationship between different types of microorganisms. They're going to be competing for the same resources, and they can prevent the overgrowth of the other by competing. We've already talked about the fact that many produce antibiotics that affect a different species to prevent their growth, so that it's a growth advantage if you're a microorganism and you're producing an antibiotic that will prevent the growth or even kill some other different organism, then that's a tremendous growth advantage. They prevent the establishment or overgrowth of pathogens. That's an extremely important point. For example, in the vagina, the normal pH is fairly acidic, 3.5 to 4.5, and that prevents excessive growth of Candida albicans. Candida albicans is a fungal organism, a yeast, which means it's a single-cell fungal organism. It grows as single cells, and it can, and it's present all the time. It's present on everyone, vagina or no vagina. It's present on males and females, on the external and internal surfaces, but it's held in check by other normal microorganisms that are growing there. But sometimes it can overgrow. In the vagina, you can get in the vagina with, if there is normal low pH, that prevents the excessive overgrowth of Candida albicans, not Canada, but Candida albicans, which can cause candidiasis, a type of overgrowth that can lead to vaginitis. Another example of organisms that are present but are inhibited, their growth is inhibited, is with Clostridium difficile or C. difficile, which is normally inhibited by the other microorganisms living in the gastrointestinal tract. However, if a person is treated with a lot of antibiotics, that'll kill off all the other normal organisms and therefore reduce competition and allow for overgrowth of, if they're carrying C. difficile, it'll allow overgrowth of C. difficile, causing diarrhea and maybe severe colitis. So you can see that this kind of idea of microbial antagonism is extremely important, that the normal commensal organisms, at least commensal for us, right, that are growing on us, are on our surfaces, they play an antagonistic role against other microorganisms that are potentially pathogenic. Now, some of these organisms in our environment can act as opportunistic organisms. They can cause opportunistic infections. These are infections by a microorganism that's not normally pathogenic, and they can occur because of a change in the local environment where the organism resides. The microbe might gain access to a new niche or area in the body, a new environment in the body. For example, it could invade through, for whatever reason, it could get in through the tissues, and you can get E. coli in the peritoneal cavity. Or or it could be in an inappropriate area like the bladder, which is external to the body but should not contain any E. coli. That's actually the major cause of urinary tract infections, E. coli infection in the urinary tract, and particularly the bladder. Sometimes Streptococcus or Staphylococcus organisms on the skin can enter deeper into the skin tissue and cause infections, and can be the cause of what's it called? And can be the cause of cellulitis, which is quite a serious infection. Sometimes there's a change in the host. The resistance of the host is decreased because of disease or some immunosuppression due to age, associated with age, or maybe because of therapy that there was person's receiving, which causes immunosuppression, and they get opportunistic infections by organisms that normally have no pathogenic effect. The organism Pneumocystis jirovecii, which used to be called Pneumocystis carinii, is everywhere, all around in the environment, but never causes any problem. Normal people would never get an infection with this. It does, however, commonly appear at later stages in AIDS patients when they're really severely immunosuppressed. Now, the idea of normal microbiota in the host has actually gotten quite a bit of attention in the last, say, 10 years, 15 years in terms of probiotics. You see a lot of advertisements for probiotics. This idea actually was first proposed by a physiologist, microbiologist, scientist named Eli Metchnikoff. Metchnikoff, in fact, was one of the fathers of modern immunology. Metchnikoff and another early immunologist by the name of Paul Ehrlich, also a father of modern immunology, Paul Ehrlich, they shared the Nobel Prize in 1908. Paul, I hope I spell his name correctly. I think it's like this, Ehrlich. Paul Ehrlich was one of the first to describe the specific immunity. He actually described, without naming it, the naming came much later, but he described the idea of B lymphocytes, and Eli Metchnikoff described the idea of a non-specific innate immunity. Those are the two main branches of the immune response: specific or adaptive immune response, and non-specific or innate immune response. So Eli Metchnikoff described the innate or non-specific immune response, and the two of them actually shared the Nobel Prize in 1908 for their discoveries. Another thing that Ehrlich contributed to was the idea of probiotics. He was actually quite a strong proponent of probiotics. The idea that live bacteria applied to or ingested into the body can have a beneficial effect, which they do. How effective these probiotics are that they sell over the counter is more questionable. You might be just as well off eating some yogurt and getting the same kind of organisms in you. Nevertheless, what normal microbiota do provide to an individual is they colonize the person and prevent potentially pathogenic organisms from getting a foothold. So they compete quite strongly with those organisms and prevent them from growing in any large number or having or causing a disease. Now, It's important to understand the contribution made by this person. What he proposed, what he proposed were cautious postulates. That's a very important concept because these are the requirements that need to be proven or fulfilled to prove that a particular microorganism is the cause of an infectious disease. You know, whenever a disease process is identified and appears to be infectious because it's spreading between individuals, and there's many ways that these things can spread, but in order to really prove that that is the cause of the disease, and often, you know, there are all kinds of proposed causes of a disease before it's eventually proven, you need to fulfill particular requirements. The requirements are cautious postulates. You have to fulfill cautious postulates in order to prove that a particular organism is in fact the cause of a particular infectious disease. And what you need to do is to be able to isolate it from an animal that has been affected by the disease or is dead because of the disease. Dead is very definitive. You don't have to measure anything in a sick animal to prove that it has a disease. If it's dead, it's certainly being affected by the disease. Which, by the way, highlights the fact that it's important for every disease to have an animal model so we can prove or investigate all sorts of things. You can't do that in humans typically. Well, cautious postulates involve isolating an organism from an animal affected by the disease and growing them in pure culture. You have to grow them in pure culture, and that way you can identify them and do tests on them, look at them under the microscope with different staining techniques. You then inject them into a healthy animal, and that animal has to come down with the same disease. So the disease sign and symptoms need to be the same in the next animal or set of animals injected. And then you have to be able to isolate the exact same organism from that animal that died due to the disease, and again identify that it is exactly the same. So you need an animal that has the disease. You need to be able to isolate the organism from that animal. You need to grow it and inject it into another. I'm going to show you, these lines are ridiculous. I'm going to show you how to draw a mouse. It's very easy actually to draw a mouse. Let's make it this color. A mouse is not a difficult thing to draw. You draw an 8, a slanted 8, like that. And then you put in the head and the body of the mouse, and there you have a mouse. So you need to have a sick or dead mouse that has the disease. You need to be able to isolate the microorganism from that animal and grow it under a microscope, grow it and examine it in cultures and under the microscope. Then you need to be able to inject it into some other mice and get the same disease process, and then I didn't draw that mouse properly. It's like this. You draw a straight line, and then you draw an arc, and then you draw the figure 8, and you put in a head, and you have a mouse. That mouse is way better than this one. Let's see. Here we go. Yeah. Oh yeah, I forgot the tail here. There you go, there's a mouse. So, mouse has the microorganism. You isolate the microorganism from the mouse and you grow it and identify it in the lab, and then you inject it into other mice, and they have to get the same disease and die, and or at least have the same disease. And it doesn't have to be mice, it could be some other animal. And then you can you have to be able to then isolate the same organism from those mice or animals that got sick. Cautious postulates. I hope that was clear. Okay, how do we classify infectious disease? Well, we do it like any disease, the difference being that it is infectious, but we look at the signs and symptoms. Right? A sign is a change in the body that can be measured or observed and is a result of the disease. And a symptom is a change in the body that is felt by the patient as a result of the disease. So when a person has symptoms, which they can feel, they may seek help. Medical help, and the person they go to looks for signs, particular signs associated with their symptoms. Okay, the person feels the symptoms, they can't feel signs. Signs have to be measured. Vital signs like temperature, respiration rate, pulse, blood pressure, things like that. You know, people look for particular signs in order to identify what kind of disease process a person has. Overall, when you see a particular set of signs and symptoms are typically accompany a particular disease. So the different diseases are characterized by a particular set of signs and symptoms. We classify disease or infectious disease as communicable if it spreads from one host to another. Contagious refers to the ease of spread from one host to another, how easily it spreads. Is it a contagious disease or highly contagious disease? Is it a communicable disease? And if it's a non-communicable disease, then it is not transmitted from one host to another. So infectious disease is always communicable in some way. It is always transmitted from one host to another. Otherwise, the disease would disappear as an infectious disease. It would run out. If it's in only one individual or a few individuals who cannot transmit it, then it's not communicable and it will not continue as an infectious disease. There are many non-communicable diseases that are not infectious. They're not due to microorganisms. But if it is due to microorganism, it has to be communicable, and it also typically would be a contagious disease, but how contagious is relative. Some are more contagious than others. All right, now in terms of the occurrence of disease can be looked at as occurring with a certain type of incidence and a particular prevalence. The incidence are the number of new cases in a given time, say per year, how many new cases are occurring per year, what fraction of the population is developing the disease, whereas prevalence is a different way of looking at it, and that's the fraction of the population at any time that has the disease. So some of the people in a population may have gotten the disease 10, 20, 30 years ago. They still carry it, the infection. And others may just be developing in that year. So prevalence is always, of course, typically higher than the incidence. Diseases that occur only occasionally in a population are called sporadic. If a disease is constantly present in a population of people, then it's considered to be endemic. And endemic, there are many endemic diseases that are always present. Not in everybody, just in a certain fraction of the population. They have a established, steady prevalence within that population. and it's constantly present. Epidemic disease is acquired by many individuals in a given area over a short period of time. Whereas pandemic, and I believe the definition of pandemic is present on more than 2 continents. So pandemic disease is infectious disease is considered pandemic if it's a worldwide epidemic occurring in more than 2 continents in many individuals over a short period. And then there's, in terms of the occurrence, there's also something that develops with the occurrence of disease and also with vaccination, which is kind of like vaccination really is sort of artificial exposure in a safe way to a disease. So herd immunity can occur if enough individuals in a population catch a disease and have recovered, and a significant percentage of the population, and I do mean a majority of the population, have had the disease, then there is what's considered herd immunity. But that's the very hard way of developing herd immunity in a population. That's the way where anyone who is immune had the disease, got sick, usually, not always, but often got sick, and some often die because of it. Depends on the disease, of course. But the other safe way of developing herd immunity is through vaccination, an artificial way of exposing people in a safe, artificial way to the disease organism and developing immunity in the population. Well, the severity or duration of diseases can be described in a number of ways. Acute disease is when symptoms develop very rapidly. Chronic disease, disease progresses slowly. Subacute disease, the symptoms range between acute and chronic. And in a latent disease, there are long periods with absolutely no symptoms at all. In fact, for certain latent disease processes, viral latent disease processes, you don't see anything. The organism is there, but you see absolutely no symptoms whatsoever. Okay. As I was saying, the extent of infection in a host is important. How much or in what location the host is infected, because infection could occur in different parts of the body. It could be limited. A local infection is very limited to a small area. Systemic infections are throughout the entire body. Sometimes you hear the term focal infection. What it means is that there is a systemic infection spread throughout the body that began as a focus. It begins as a localized infection or localized site of infection, and it spreads from that focus. So the focus of the infection is there. It spreads throughout the body. Sometimes they treat the person, and the disease or infectious process seems to have been successfully treated, but then it reoccurs because the focus is still there. And that's a focal infection. Bacteremia is a term referring to the presence of bacteria in the blood. And septicemia, which is quite serious, is when the organisms are actually growing, dividing in the blood. Toxemia is the presence of toxins in the blood, and viremia is the presence of a virus in the blood. Again, in terms of extent of host involvement, You can have primary infection and acute infection causing the initial illness or disease. Secondary infections occur following a primary infection, where the primary infection is the initial process that makes the person susceptible to the secondary infection. So if a person, for example, I guess one example would be infection with HIV, which causes immunosuppression, leading to pneumocystis jirovecii, pneumonia. So you get an opportunistic infection followed after a primary infection. And then there's subclinical disease, where you have no noticeable signs or symptoms, at least initially, can be for quite an extended period. The organism is there, it is growing, it is causing some changes and damage, but it's subclinical without signs or symptoms. And that's true for HIV, hepatitis A. Are there predisposing factors to a person's susceptibility to infectious disease? Yes. Urinary tract infections are more common, much more common in females, because they have in other words, they have an increased susceptibility because they have a much shorter urethra than males. And therefore bladder infections, urinary tract infections are much more common in females. Sometimes people have particular genetic traits that make them more prone to getting an infectious disease. For example, sickle cell anemia. Climate and weather can contribute to increased susceptibility. Certainly fatigue causes and stress causes increased sensitivity to or susceptibility to catching an infectious disease. Age is an important factor, and age is a tricky one. Sometimes it's the very young and the very old who are more susceptible. Sometimes the young are not susceptible and it's only the older individuals. Certainly there is a clear, very well documented decrease in immune function with age. So with age, as people get older, they become somewhat immunosuppressed and therefore more susceptible to infection. Lifestyle is very important. Poor nutrition, poor habits, smoking, etc., can make a person more susceptible to infectious disease. Their occupation. And then treatments. Drugs can contribute to problems. Some drugs certainly do cause immunosuppression and an increased susceptibility to infection. So these are predisposing factors that can increase the susceptibility to infectious disease. Infectious disease has a period of a clear sequence of stages, and some can be this is a general graph, but a general presentation of the ideas, but some of these stages can be extremely long or very short, depending on whether it's acute or chronic or latent or persistent infection. There is always, with infectious disease, an incubation period. And during that incubation period, there's no signs or symptoms at all, but the infectious agent organism is multiplying. You can see the number of microorganisms is shown here along this axis, and time along this axis down here. So what you have is an incubation period with no signs or symptoms. And it's an important concept because you want to be able to identify this period. It's called the prodromal period. You want to find the characteristics that can be seen during that prodromal period. These are mild signs or symptoms, and if you can identify those, then you can find what it is you need to look for to try and identify the infectious disease early on before it starts to really cause clear, apparent signs and symptoms of the disease process. So catching an infectious disease early is important. It allows treatment to start much earlier, and even a few hours can be very, very valuable if you catch it in time. So the prodromal period, the signs, mild signs and symptoms that are present in the prodromal period are very important to be able to identify. Then, of course, there is that period of illness where the disease is full-blown. You see all the signs and symptoms, and it may result in death. However, it could also result with treatment or sometimes without treatment, but this depends on the disease. Without treatment, the period of illness would be prolonged. Typically, with successful treatment, it's shortened. Death, it's also shortened, but not successful. So the next period is the period of decline. If the person survives and is getting better, then there's a period of decline in the severity of signs and symptoms, and then a period of convalescence where they're getting over it. So an incubation period, a prodromal period, and the period of illness. And then when they're recovering, a period of decline, period of convalescence. The incubation period, that initial period during which the organism is increasing in number, really depends on how long it is. How long that period is really depends on the nature of the particular microorganism that causes the disease, its virulence, how many have infected the individual. That can be extremely important. If it's a smaller number, it may not infect the person at all. If it's a large enough number, it could infect them, but take much longer to develop into causing signs or symptoms. So the number is very important, and the degree of host resistance is important. All of us differ one from the other, one from the next. We're not identical, and each person's resistance can be different from the other, and you can get quite a range in terms of susceptibility. So, what allows organisms to spread? Spread of infectious disease is a key feature of the disease. They have to be able to spread. So what allows them to spread? Well, one thing is there has to be some sort of there has to be some source or reservoir for the infection. There has to be a continual source of infectious organisms, either human or non-human, could be human or animal, or even a non-living source of that organism. Human reservoirs, these are called reservoirs of infection. Human reservoirs can involve carriers who show no signs or symptoms but are carrying the disease, the infectious microorganism, and can infect others. You can have inapparent infections or latent disease that can inapparent infections that again can act as a reservoir and help spread the disease. There are a number of diseases that don't have a lot of strong. There are stages where you don't see many signs or symptoms, and they are infective. They can infect others. AIDS, gonorrhea, hepatitis can all act that way. Another type of reservoir, non-human reservoir, are animals. Animals form an important source of many diseases, particularly new diseases. But there are many diseases that are carried by animals that can cross over into humans. Two examples are rabies and Lyme disease. Diseases that primarily occur in animals but can be transmitted to humans that humans can catch are called zoonoses. And zoonotic infections are something that can be a problem and people need to be aware of. There can also, besides human reservoirs and animal reservoirs, you can also have non-living reservoirs. And examples of diseases where there's a non-living reservoir of the organism would be botulism, which is caused by Clostridium botulinum, and in which the organism is found in food that's been contaminated with it. And it can be canned and sealed. It's an anaerobic organism, so if it's due to improper sterilization of the food when it was canned or bottled or stored, then it can be a non-living reservoir for that. It doesn't take too many organisms to actually produce enough exotoxin to be to have really terrible symptoms with food botulism, food poisoning. Tetanus is another one that can have a non-living reservoir. It can be in the soil, the organism, Clostridium tetani. Another example would be Legionella pneumophila, the organism that causes Legionnaire's disease, which can grow in water that's accumulated and sitting still, like in a pan underneath an air conditioning unit that might spread it throughout a building. So there are a number of different things to be aware of in terms of reservoirs, human, animal, and even non-living are all important. And here's just some examples that you can, for your own interest, look at, but we've talked about some aspects of these, and here's some others. There's a lot of them, and it's these are all actually here, these are zoonoses. Selected zoonoses for bacteria, viruses, fungus, protozoa, and helminths, worms. All right. I think that's enough for now. That would be the end of lecture 14. That would be not the end of lecture 14, but lecture 14 part 1.